Okay. Thank you, Angel, for your kind introduction. It's, of course, a great pleasure to be here again and talk about the evolution of chemical science in TIFR. It's, of course, a great challenge because chemistry, as all of you know, is a subject which actually is quite encompassing and goes all over all kinds of topics. And certainly, I cannot claim that I can do justice to everything of that one. I will try to do my best and cover the history and try to cover the extent to which I can understand the current research which is going on. So can you start this, please? Yeah, excuse me. Nice. So this is the pointer. Yes, okay. So I have tried to collect information from the archives as much as I could, and I also tried to collect information from the colleagues in the department and try to put it together in a compact form. And of course, being the last speaker of the session, I can possibly go on till the till the cultural program. But I will try not to do it. There is a dance program. Okay, here it is. So we have this, as I mentioned to you, the chemistry lies here in the sphere of knowledge. You have the maths here, physics, chemistry, and biology. It has an interface. Chemistry has interface with both chemistry and, oops, what happened? Huh? Okay. Okay. So you have this interface with chemistry. Chemistry has interface with the physics on one side, biology on the other side, and maths has interface with everything. So you need to have all of them. So you need to have math, physics, chemistry, biology, all put together to make it a good science. So research focus at TIFR has been continuously changing because you have to remain in the forefront. As the science develops all over the world, you have to keep changing yourself, look for new projects, look for new problems, so that you're always at the frontiers. Members of the group regroup. So because as the subjects change, you'll have to regroup yourself, put together the expertise, and suddenly you can solve a consolidated problem. New faculty are recruited considering the emerging fields. So this has been the strategy which has been adopted at the department and of course probably at the institute as well. So this is a successful story and I will try and give you hints of that kind of activity in the next few slides. Okay, the beginning is of chemistry at TIFR. This is the statement from Homi Bhabha, the visionary he was. And he wrote here, nuclear magnetic resonance is not, in the strictest sense of the word, an electric paramagnetic resonance, he said, certainly not nuclear physics. At that time, nuclear physics was the main theme of TIFR. And these are, however, techniques with great impact and application in chemistry, solid-state physics, and biology. It is therefore an appropriate field for the institute to consolidate and develop. And Dharmati has the ability to and drive to lead the program. Is certainly a field with great promise, and Dharmati has in his field very wide and extensive international contacts, starting with the close contact with Felix Bloch. These relationships are of great importance for himself and his students and thus for the institute. There is a history here that this is Pandit Jawaharlal Nehru, of course Homi Bhabha, and this is Dharmati here. Homi Bhabha got Dharmati from Felix Bloch, and Felix Bloch had won the Nobel Prize. Well, he had not won the Nobel Prize yet, but he was the discoverer of the nuclear magnetic resonance in the liquid state. And Dharmati was associated with Felix Bloch. We brought him here. And then he went there, of course, he did something fantastic things. And that was the initiation of chemistry in TIFR. See here, this is the classic paper of Dharmati, which shows the discovery of the chemical shift. Chemical shift is the crucial element in magnetic resonance, NMR, nuclear magnetic resonance. Dharmati wanted, Felix Bloch wanted to measure the moment of tritium, and there was this student, Arnold, who was actually working on that one. And they were supposed to measure the magnetic moment. They thought magnetic moment of tritium or proton or whatever that is going to be something. And there will be one absorption line for in the resonance phenomenon. And Dharmati said, well, why not we put water? They said initially they put water, H2O, and want the tritium from there. But Dharmati said, for what reason one doesn't know, he said, let us put alcohol. So he put alcohol in the magnet. In the magnetic NMR experiment, the NMR experiment consists of a magnet and then of course the receiver where you put in the RF coil and they get the signal. So he put alcohol there. And when he put alcohol, he actually saw three signals. See here, CH3, CH2OH. He got three signals. Felix Bloch could not believe this. He said, how can you have three signals for the proton? The proton is same. So its nucleus is the same. So how can you have three different signals? But there was a lot of debate, and finally they concluded, okay, it is something to do with the electronic environment around the proton. Proton is not a bare nucleus, it has electrons around it. So different chemical environments can be there, which can cause different chemical shifts or resonance absorption frequencies, and they got these three different lines. And so in this, CH3 belongs here, CH2 is here, OH is here, and their intensities are in the ratio of 1:2:3. This is precisely what happens when you have the number of protons different and all resonate at the same frequency. You have these three different lines. So this is the classic paper. This was the beginning of the chemical shift and entry into chemistry of NMR. And then at that time, so when Dharmati came, actually they started a group here, which was then called as magnetic resonance, but it was called as nuclear and electron magnetism. It was called the NEM group. Professor Vijayaraghavan was already there. Dharmati, Kanekar, Vijayaraghavan, Venkatraman, L.C. Gupta, S.K. Malik, G. Govil, and so on, so on, so forth. Nagarajan is also here, and there were many more. So, and therefore, with this, actually, they started this activity here, and this was called as the nuclear and electron magnetism group. And unfortunately, Dharmati died in 1963 because of he had a heart attack somewhere. So while giving it, and then of course, some differences developed, and then they split this group into two. One was called the chemical physics group in 1967, and Arun, of course, you can correct me if there are any errors in the in the history. And solid state physics group, this was Vijayaraghavan, Gupta, S.K. Malik, and so on, Nagarajan and so others. And then here's the magnetic resonance we had here: Kanekar, Venkatraman, Govil, Khetrapal, Prabhananda, M.M. Dhingra. And magnetic susceptibility, this was an area which was followed by S. Mitra. The Masbour effect, which was S.K. Date. Electrochemiluminescence, K.S.V. Santhanam. And microspectroscopy, Doreswamy and S.D. Sharma. Theoretical chemistry, theoretical biology, it was B. Venkatraman, G. Govil, Anil Saran. These were the people who were actually working in these areas. In those days, many of these people were actually coming from the training school. They would join the training school, then of course they top the training school, then they come here. Likewise, actually, Professor Govil came from there. And there are also others in the physics arena. And the emphasis at that time, during the first 25 years, I tried to collect this information from the archives and also from the book which was printed in the 1970. So the 25 years of TIFR, and the emphasis was on instrument building and using as much as possible, try to build instruments to be on the front line. And this was the first NMR in the country. This was the Varian machine, and Dharmati, Bhabha got it for Dharmati. This is the 60 megahertz NMR machine. Varian high resolution NMR spectrometer. Those are the only machine of its kind in the country at that time. There was no other NMR machine in the country. And then you have what were they doing with this? They were actually recording the spectra of organic, inorganic small molecules and molecules oriented in the limmatic phase. This limmatic phase, this was the liquid crystals. This was the Khetrapal's work. CL Khetrapal was also present here at that time. And Govil and Professor Govil and Professor Kanekar, they were interested in measuring the chemical shifts and the coupling constants with other parameters, which we will not talk into the details of those. They were also actually calculating the NMR spectra from quantum mechanical methodologies. So simulating the spectra, comparing with experimental spectra, and try to extract these parameters of chemical shifts and coupling constants. So this was the instrument at that time, and this was an electromagnet here, and this is the control. Electromagnet requires huge amount of chilled water supply to cool the magnet, otherwise it will become hot, and then of course it is not stable. Then all sorts of aberrations will happen in your spectra. And this was another instrument which was built here. This was the Gaui balance. Gaui balance, it's called. It's used for the magnetic susceptibility measurements. And with the magnetic susceptibility, you can determine the electronic structures of transition metal complexes and intermetallic compounds. Of course, magnetic susceptibility is very crucial in magnetic resonance. You can measure the magnetic susceptibilities in every molecule. In those days, of course, they were actually measuring on the transition metal complexes and intermetallic complexes. Samaresh Mitra was the primary faculty during that time. And then the ESR spectrometer, this was Professor Venkatraman's baby. So they were actually they built this instrument using electromagnet here, and they were using this to measure free radicals. ESR means electron spin resonance. You need a free electron here, and the electron spin is used as a magnetic resonance probe. And you have ESR spectrometer. They were measuring C13 hyperfine splittings in free radicals and photochemical and kinetic studies. That was what was going on. And all this, look at the instrumentation here. It will occupy the whole room. If you see today's spectrometer, they actually sit in a small corner. So this is occupying a whole room. And they were, this is all built here in this institute. And this was done by Professor B. Venkatraman and Professor Prabhananda. Prabhananda actually shifted later on to some other areas, but in those days, actually, he was with Venkatraman, and primarily he was working on the ESR spectrometer. And this is Doreswamy and Sharma. These again people came from BRC. They were working on microwave spectrometer. This was the initiative from Professor Venkatraman, and they said they work on a rotational spectra of molecules. Incidentally, I remember people talking about something like astrochemistry. What were they measuring? They were actually measuring the rotational spectra of small organic molecules. Maybe they are present in the galaxy or somewhere. So whether the spectroscopy can help in detecting any of those ones. So therefore, they were actually doing rotational spectroscopy, and the instrument of the microwave spectrometer was built here. And the molecules in a gaseous state, dipole, and quadrupole measurements. This was Doreswamy and Sharma. And then they had this another X-band ESR spectrometer, the rotating magnet. And this was actually so that you can rotate the magnet for oriented magnetic field in a particular direction you want. And this was used to measure relaxation times. So this was done by Professor Venkatraman and Professor Prabhananda, and they were using this to measure spin lattice relaxation times in the microsecond region of organic free radicals and anisotropic studies in the solid state. And look at the effort of the instrumentation here. And this is the electrochemiluminescence. This was Santanam. Santanam was actually measuring the emission during electrolysis from photoexcited molecules, the heterocycles. So this was used to understand photochemistry and that happens in your molecules. And the instrumentation was built in this year. So the emphasis, why I'm showing all of this is that emphasis at that time was to build your own instruments as much as you can. So you choose your projects, choose your problems, of course, build your instruments to be on the forefront. However, of course, they could not last for too long. That's a kind of a tendency. If you have for too long, then so surely you are getting lost to the commercial market and you are not able to produce outstanding science. That kind of a thing actually came in the way. Okay, and then that was the experimental work, and then also this area called theoretical biology. And theoretical biology here, what people were trying to do is predict shapes of peptides, proteins, nucleic acids, and lipids by calculating their energies using various methods. And this is, of course, you use your different kinds of methods were used. One of these called the classical potential functions. This was initially started by G.N. Ramachandran in Bangalore. He used classical potential functions to calculate the energies of a dipeptide to find out the conformations of peptides find out the minimum energy conformations. And then Govil and Saran, of course, they used different methods. They used quantum mechanical methods. These are approximate methods. These are not rigorous quantum mechanics. These are actually quantum chemical methods, which are lots of approximations, various kinds of approximations in the integrals. Of course, the integral approximations are also based on certain experimental data. So you use these methods, complete neglect of differential overlap called C and DO, intermediate neglect of differential overlap, I and DO. These are SEF methods, okay, the variation principle methods. But then, of course, you have this one more. This actually will take very time consuming. At that time, you used to do these calculations in a computer called CDC 3600. So it was such a slow computer by today's standards that you submit your data with a punch cards, you would get the results after 2 days to find out that there is a mistake in the format of your data. So, and then you submit it again, wait for two days again, and that sort of things used to happen. This is a very painful exercise to do these calculations, but nevertheless, they were quite successful in achieving this. And this was the PCIL or perturbative configuration interaction using localized orbitals. This is the perturbative methods, relatively faster, and you can do it relatively easily. And search the conformational space or minimum energy conformations shapes play important roles in biological function. Of course, after all, why do you want to determine the shape, the conformations? Conformation means the geometry of the molecule, how does it look like, what are the active sites in there, where the interactions happen, all of this is very important. So for that, you need to do the conformations, and these are important for biological function. This was primarily followed by Govil and Saran. And so this is the kind of a approach. So this was the Ramachandran map. This is also called the Phi-Psi map for the peptide. You have the peptide going like this. You have particular torsion angles here which are crucial. This is called the phi torsion angle. This is the psi torsion angle. Is the relative magnitudes of these torsion angles which determine the geometry of the whole molecule, the shape of the molecule, peptide. And you have this also called as Phi-Psi maps or Ramachandran maps. Orange regions indicate the allowed regions, or rather one should say this, if you are anywhere here, they are not allowed because they are sterically out, they overlap. If there is a steric contact force, it is not allowed. So therefore, if you get a molecule which has phi and phi torsion angles in this area, it is a disallowed conformation. If anybody, even today, if it determines the structure by whatever method, crystallography or NMR or whatever, if you get a value which is falling here, is incorrect. You'll have to go back and look at it once more, or find a justification why it has happened under those conditions. So therefore, this is a very, very fundamental discovery. Actually, many people felt that, okay, G. N. Ramachandran should get the Nobel Prize for this. But it so happens that, okay, you don't get everything. And then based on these angles, you have the different kinds of structures for the proteins. This is called the primary structure of the protein, which is the sequence of the amino acids. And as you know, there are 20 different types of amino acids. You connect them one after the other here, and then you form a polypeptide chain. This forms the primary structure, and these ones can arrange themselves in a particular manner to have certain interactions here. These are hydrogen bond interactions between two chains. One chain is running like this, other chain is running like that, and you have interactions here which will hold them together. This is called the beta structure, and here it is a chain which is running like a helix, and the interactions here actually hold the molecules together. It provides stability to the molecule. This is the helical structure, and then of course you can have combinations of these to form a tertiary structure where your helices and beta sheets together. And then of course the structures of such can organize themselves in a more elaborate way that's called the quaternary structure. So you can have structure defined at different levels, and all of these are important to understand. And similarly on the nucleic acid side, nucleic acid is your genetic material. Typically one knows that it is a double helical structure. You have two strands running here, go in the opposite directions, and this is called the bDNA. And the later discovery in 1979 was the zDNA, which is the left-handed DNA. So the chain runs in the left-handed manner here, whereas these are right-handed helices. And two chains are held together by hydrogen bonds between base kind of a base pairs here, and that is the genetic code. So the sequence of the bases is important, and that is your genetic information. So we'll not go into all of those details, but at the fundamental level, the structures of these, the backbone structure is determined by these kinds of torsion angles here. This is the molecular structure of the DNA. You have the sugar ring here, the phosphate, phosphodiester bond here, group here, then there's the sugar here, and then it goes on like that. And at this point, you also have a basis attached. There are four kinds of bases, which are called as A, T, G, C. The sequence of this basis is the genetic information, and these bases pair themselves here, A pairs with T, G pairs with C, and that is what is responsible for the replication process. Okay, and here these are so-called the isoenergy contours. So what are the allowed energies, allowed conformations, and you plot here two particular torsion angles. This is omega and omega prime. You plot the two torsion angles here, and what are the allowed conformations? You see it is only this much area which is allowed. Not even this actually is 5 kilocalories or 3 kilocalories per mole. You have very little populations. So if it has to be allowed, it has to be below 3 kilocalories per mole, and you have mostly in this area, this sugar geometry is, the phosphodiester geometry is allowed. So these are the values which are present in all of these. It's written here, the structures, all RNA structures, so the DNA A, DNA B, they are all present in this area. So this was determined by the classical potential functions. Okay, there was also analytical quantum chemistry, which was happening. There was a theoretical biology, there was theoretical chemistry. We also had here theoretical chemist, Ramakrishna Ramaswamy, and he actually collaborated with Deepak Dhar in theoretical physics. And he was actually a non-linear chaos process, so chaotic process. So he was actually interacting with Deepak Dhar here. And I also happened to do something of analytical quantum chemistry here. quantum mechanical theory for transport across biological membranes. The idea was to choose the membrane as a potential barrier and calculate the flux through the potential barrier. And interestingly, this was used by some other group in Spain. They derived the laws of thermodynamics, irreversible thermodynamics, on the basis of this theory. So that was something which is quite satisfying. Okay, so that was for the first 25 years. And what happened next? After 1975 onwards, this research areas continued, but actually took a different shape. And now, because the technology had developed, you had better equipments, more equipments, all kinds of things happened. Then the research could be classified into these particular areas: molecular biophysics and structural biology, bioinorganic chemistry, chemical dynamics, materials chemistry, electrobioluminescence, biochemical fuel cells, molecular electronics, theoretical chemistry. I've colored them here, and the same kind of colors are present here to identify the faculty who are doing in those areas. I don't want to go through the whole list, otherwise it will take a lot of time. And certainly I will not be able to describe achievements of all of these people in every area. Here my objective is to sort of list the kind of areas, how the progression has happened, progression of science has happened, progression of chemistry has happened in the department, and highlight some of those things which are, well, not too many, because otherwise I have a danger of leaving out some of those in the process. Nonetheless, I will just say that the techniques which were used are magnetic resonance, laser spectroscopy, mass spectrometry, calorimetry, circular dichroism, AFM, TEM, IR, Raman, etc. And the department has made pioneering contributions which have enriched our knowledge of basic chemistry and chemical principles of biological function. And once again, there was an emphasis on instrumentation, building. Indigenous equipments have been built for supersonic jet spectroscopy. This is actually the work of Sanjay Vatagaonkar, and he is not here. He has his daughter's wedding recently, so he's quite busy there. Fluorescence correlation spectroscopy. This was actually the one which was built by Sudipto Maiti, who is the current chair of the Department of Chemical Sciences. Multiphoton imaging also was from him. The T-jump was actually from Professor Prabhananda and G. Krishnamurthy. GK is here. And these were done for the kinetic measurement, to do the chemical kinetics, study the kinetics of enzymes. And to name a few major ones. Again, then of course, the National Facility for High Field NMR, established as members of the department, is now an acclaimed international NMR center. This was one of the major achievements. Actually, it served not only the Department of Chemical Sciences, but the whole country in a big way. So I would like to spend a little bit of time here explaining these ones, how it has gone on. And the Chemical Physics Group was renamed as Department of Chemical Sciences or DCS in 1997. You know, the department has been going through review processes as other departments. Actually, the whole institute is going through the review processes, and every now and then, of course, there are inputs from the various reports, and these actually have influenced also the choice of the projects for the subsequent years. And so evolution of NMR at DCS, it started, as I said, from Dharmati, and the Professor Vijay Raghavan's group went into the solid state physics group, and there it was the wide line NMR, and L.C. Gupta, Sampat Kumaran, Grover, these people are working in the wide line NMR group. And I'm not dwelling into that one. I will focus mostly on the things which were in the chemical sciences, which is mostly high resolution NMR here. So high resolution NMR started with Dharmati. Then the first 500 megahertz spectrometer was installed in TIFR in 1983. And this is a big contrast to the 60 megahertz magnet which was there, which I showed you with earlier. 60 megahertz magnet was an electromagnet with a huge, occupied a huge room. And this 500 megahertz is a superconducting magnet, stays in a corner like this, and your console sits just adjacent to it. Then, of course, the whole technology change was there. Of course, you could not have kept pace with that sort of a technological development. And then regularly the system was upgraded to keep it state of the art. Then in 1995, a 600 megahertz NMR spectrometer was installed. And why did this happen? Look here. The research focus has been changing. You are initially with small molecules, 10 atoms, 20 atoms, 30 atoms. And slowly you are moving towards complex large biomolecules, proteins, nucleic acids, membranes, in vivo, metabolomics, the whole area which was being developed. And therefore, they required these sophisticated equipments, and these were acquired by TIFR. Of course, I must say that the government of India has a great support for this facility. all the Department of Science and Technology, Department of Biotechnology, CSIR, all of them came forward to support this and funded to installation of all these spectrometers. Upgradation of the 600 megahertz console, installation on 800 megahertz spectrometer with a cryoprobe, and then of course in 2006, a new building was built to accommodate all of these spectrometers that is standing today as National Facility for High Field NMR. And the people who are associated with that, they are all listed here. Of course, we need not go into the details of all of those names here, but they are there for the record whenever somebody wants to see this. Okay, so this was the first 500 megahertz NMR spectrometer. You can also see outside on the display there. People are trying to identify what this photograph is. So I said, okay, this is the NMR spectrometer. Unfortunately, this slide is not very sharp here in this. So, but this is the magnet. This is the 500 megahertz superconducting magnet. You compare this with the 60 megahertz spectrometer, which I showed you in the beginning. And this is the console. So therefore, absolutely clean. And once you charge this magnet and you put liquid helium, keep on putting liquid helium, you don't have to worry about anything else. The temperature remains the same, absolute perfect homogeneity. You have the homogeneity of the sort of 1 in 10 to the power 9, sometimes 1 in 10 to the power 10. That level of homogeneity you have it, and therefore you can get very sharp lines in your NMR spectra. And this is the NMR facility, which was new building which came up in 2006. And with this, we could accommodate all the spectrometers here. And now this is the inauguration of this building. This you have here JJ Bhava, and of course all the big people are here in this. You can see Shovo here, and Professor Vijayaraghavan who is here. Shovo inaugurating this. This is Professor Khetrapal. Then you have Professor Govil here, and the other members. And Anil Kumar is here. From all over the country, people had come for the inauguration process. And this is the 800 megahertz. This was the highest field available at that time anywhere in the world. And this is the pumped magnet, and this requires very careful management of the NMR system. The technology is still developing. We should continue to maintain the technology and keep it state of the art. And it was very interesting to see JJ Bhava was so curious to know what was happening, and so he is explaining to him. But then we also explained in some other pictures later on. And this is one in the same picture a little bit. This is the 600 megahertz. Now it is inside there. That was also inside. This is 800 megahertz spectrometer. Now this is a 500 megahertz wide bore magnet, because this now we entered into the solid state area, solid state physics, solid state NMR, and this was the wide bore spectrometer which was obtained. The success of the national facility prompted DST to suggest expansion. What I mean success, not only the good research what you do, but also the service you provide to the rest of the country. Because we said this is the only place where there was facilities available for every researcher from across the country to come and use it. And therefore they were all coming, the service was provided, no charges, some charges, because of course you can pay some charges, otherwise no charges. So that was the service which was provided, and to serve the nation even more. And they said, okay, why don't you expand it? I said, okay, at that time, that okay, where is the space? I don't have space to expand it here. If we got more spectrometers, surely we will want to have it, but there is no space. So we actually made a proposal to make an NMR center elsewhere. And at the same time, this proposal for the TIFR Hyderabad came up, and then the land was given in Hyderabad. And it so happened that, okay, this could be shifted. This new center could be created in TIFR Hyderabad, and MK is now there taking care of all of that. And that is developed, and this has played a major role in the creation of TIFR Hyderabad. Now, this is actually a kind of a picture which shows the evolution here. All people, see, this was in the 1974. This is Felix Bloch here. TIFR organized an NMR conference in 1974. This is called International Society of Magnetic Resonance. It was held in this hall. This particular, I remember this. And Felix Bloch is here. There is Schlichter is here, and all big stalwarts. They were all present here. So the TP Das was here. TP Das was a big name that time. So, and Felix Bloch visited this NMR labs, and this is Richard Ernst here. And I see this Richard Ernst, Chidambaram here, Professor Govil, this is Professor Khetrapal, Shobo, and this is JJ Bhava, this is Mustansi here. Then this is Malcolm Levitt, who is actually from UK, who came here. He keeps coming here. And here you have Anil Kumar, this is Kurt Wittrich, and as you know, all of these people got Nobel Prize. Kurt Wittrich got Nobel Prize, Richard Ernst got Nobel Prize, okay, and Felix Bloch had got Nobel Prize. Switchter is here. Of course, Purcell was not here at that time. And the first announcement of MRI, which was called as gigmatography, was made on this stage. This was the Paul Lauterbur. He was giving a lecture in this platform. And this was taken at a meeting. It was a summer school which was held here in TIFR. And this is the standard place where we take pictures of all of those. And this is Kurt Wittrich here and Richard Ernst here. And at that time, of course, people were surprised at how did you get both these Nobel laureates on the same platform? Said, well, I said, they are there. So how did you manage to? I said, okay, don't ask me that question. They accepted my invitation. They have come here. We are very happy. So you be happy as well. Okay. So research at the NMR facility was, this is the physics, chemistry, biology interface. And here, Professor Vijayaraghavan, you can see here, he played a very major role in the establishment of the NMR facility. He was all supportive for us to provide any kind of help that was required. And liquid helium, liquid nitrogen, and this is Imam Sharma, who is an FRS, and this is Professor Khetrapal, Professor Anil Kumar. These are all the stalwarts of NMR community in this country. And the areas that happen here, the nucleic acid structure and dynamics, peptide protein structures and dynamics, protein folding, metabolomics, drug design, in vivo, NMR methods, solution state, and solid state NMR. And solid state NMR started with the appointment of P.K. Madhu, who is also now in the leading the NMR group at TIFR Hyderabad. Okay, now this I will just show you one of the major achievements here. We're using this. We had 5 minutes. Which slide? Okay, possibly 7 minutes, 8 minutes. Okay. So this is one of the major accomplishments, which was I just want to show you here. This is the largest protein self-assembly elucidated. This is the size of 20 megadalton. We are talking about 10 kilodalton or even less, 5 kilodalton, things like that. That is the kind of a size which one was working on. Here is an achievement which shows several megadalton size protein is associating into a several megadalton size aggregate. The mechanism of that was elucidated here using our facilities that was here. Of course, this required a lot of methods development, solution state and our methods development that was preceding this. It allowed us to calculate this and showed us how association can lead to the formation of a rope-like structure. This is by protein, which is called as dynamin, which is involved in endocytosis process and biological function. We will not go into those details, but point is that this was the first one to be demonstrated that with the protein getting folded, it is getting associated and forms of rope-like structures with the self-assembly. Okay. So in that I can probably skip this. The result of all of this, I don't want to go into the individual accomplishments, but all these people get recognitions here in the country in terms of various kinds of fellowships, with awards and things like that. We will not go into those details here, but nevertheless, the research was very well recognized by this Indian community and the international community. And this was able to do many international conferences to India, overcoming stiff competition. A SMAR meeting in 1974. This was the International Conference on Theoretical Biophysics in 1980. International Conference on Magnetic Resonance in Biological Systems twice, 84 and 2005. International Biophysics Congress, IOPAB in 1999. Symposium on Recent Trends in Biomedical Research in 1999. Symposium on Advanced Bioinorganic Chemistry 2009-2017. Of course, a large number of national conferences have been organized. Now, I come to the current faculty. The current research, last decade, as I mentioned earlier, there's a lot of people have been recruited. They are promising new areas of research. And now, the good thing is that all of their research, although they have the individual projects, they can be grouped into certain areas. They themselves are grouped, right? The molecular biophysics and imaging, and these people are involved in that. Maiti, Koti, Ancona, Dasgupta, Venkatramani, Shamla Mazumdar. Theoretical computational chemistry, Venkatramani, Voora, Vatgaonkar. Chemical biology and synthetic chemistry, Ancona, Dasgupta, Polshetiwar, Kushlani, Maiti, Patra. Bioinorganic and biomimetic chemistry, Shamla Mazumdar, Kona Datta, Malay Patra. Nanoscience and catalysis, Kushlani, Dasgupta, Shamla Mazumdar, V. Polshetiwar. Chemical physics and dynamics, you have Maiti, Dasgupta, Voora, Venkatramani, Mazumdar, Vatgaonkar. Notice that each one has his own project, but they are such that, okay, they can be grouped into others, they can collaborate. So that is a very good idea. So that you can go ahead with this contribution. The wisdom of each of these persons can contribute to the development of a great science. And indeed that has happened. And here are the individual achievements which I want to show, individual projects which they have. This is of Sudipto, as I mentioned to you earlier. He is one of the senior ones there. Sudipto Maiti and Shamala Mazumdar, these are the most senior ones of this. And Sanjay Vatangokar is already superannuated. And we developed several types of biophotonic tools to probe interactable problems. Some of these instruments have become high-tech commercial instruments here. So this actually is a high-tech commercial instrument. And Sudipta, I know, he actually conducts workshops regularly to teach how to build fluorescence correlation spectroscopy spectrometers. And he has also donated several of those to the individual groups in other parts of the country. And important publications are coming out here from this. And this is Amartya Bose on focuses on using formalisms of path integrals and semi-classical mechanics to derive computational frameworks that render specific simulations feasible. And this, we will not go into the technical details. In fact, these were the slides which were prepared by them and given to me. I just show it to you here for your benefit. You can follow it up later on to the individual exercises. Okay, this is Jyotish Mahan here. So he is engineering molecular chemistry, which drives the solar energy conversion. Okay, research group that uses time-resolved laser spectroscopy to develop new materials and chemistry paradigms for solar energy conversion and photocatalysis. So they have very important, very nice publications there. There is indicated that the photocatalytic cycle has the various steps. You start with the different molecules. Okay, you have the radiation coming here, then it generated ion. Okay, so and then you have this ion goes out. Okay, radical is created. So you have the aldehyde here. Anyway, so these are all details. And the designing new chemistry, that is agenda what they have, and this is the instrument which has been built in the lab, which of course uses lasers and so femtosecond lasers. And this is Ankona Datta. She is a chemical biologist. She actually develops molecules, identify biosensors to molecules, and this is a group there. Track essential biomolecules to second messengers in vivo. The ultimate aim is to apply the tools to visualize and track crucial small molecules and ions involved in regulation of essential life processes like cell signaling, autophagy, and neural activity. The modus operandi is to design and synthesize selective fluorescence and magnetic resonance imaging probes that can image target molecules in living systems. And this is Deepa Kushlani. She is a materials chemist. She actually joined in the late 1990s. And her objectives are here. Capture solar energy, photovoltaic cells, storage of solar energy. These are pictures of the same one, but the aims are here. Conversion of solar energy into chemical energy, formation of useful chemicals via photocatalysis, pollution remediation, conversion of carbon dioxide via electrocatalysis. So these are the specific details of the individual molecules and individual analysis that happens in these ones. She was invited to write a review article on all of these areas. Okay, now Koti. So he is actually a graduate student who is a product of TIFR itself. And of course, he diversified into other areas after his PhD. And during his PhD also, he did outstanding work. The following are the research interests: investigation of mechanical behavior and functional role of different classes of proteins. So he actually connects these molecules and puts a force, mechanical force, and how does the mechanical force affect the performance of the structure of the molecule. Engineering novel proteins with diverse mechanical functions, elucidation of mechanical unfolding pathways, and the relation with biochemical pathways. Development of novel single molecule assays for protein DNA, and protein RNA interactions. Shamla Mazumdar, of course, he is one of the senior ones, bioinorganic chemistry. He has been working initially with small porphyrin complexes, and then of course slowly went into the enzymes. Biological enzymes which contain these porphyrin rings. Cytochrome oxidase is one of the enzymes which he has been working on for many years. The metalloenzymes currently being studied are variants of cytochrome C oxidase, cytochrome P450, and peroxidases. All of these contain porphyrin rings inside, and the structures of these, some of these ones are indicated here to understand the catalysis process, the mechanism of their interaction. And he, of course, uses all kinds of techniques, NMR, high resolution mass spectrometry, and several spectroscopic techniques. And this is the new faculty. Of course, I have not met him. Chemistry under light matter, strong coupling. These optical modes are tuned to electronic vibrational transitions of the molecule to generate polaritonic states. The role of polaritonic states in modifying physical and chemical properties of the molecule is studied. So this is a picture. Actually, these are the slides which they provided to me. So I am showing this for your information. Chemistry under light matter strong coupling. Okay. So this is another chemistry under light matter strong coupling. Oh, this is the same one. Same one. So that is only half of that has come, is it? Earlier. Okay. Malapatra synthesis, medicinal chemistry, and drug development. These are the keywords here. You have the drugs, you have the anti-cancer screening, cell biology, targets, in vivo efficacy and biodistribution, toxicology, pharmacokinetics and metabolism, antibacterial screening. So what it involves: synthesis, characterization, and bioconjugation. These are the ultimate applications there. Okay, and then of course the methods are here. So you synthesize them and characterize them for the purposes what has been mentioned. Okay, when topics are here, you have the chemistry biology interface here. It's on one hand you have the diagnostics, and the other hand you have the therapeutics. And this is nanocatalysis. This is Paul Shetivar, and he is a materials chemist. Actually, he is one of those who actually created quite a name in by discovering this dendritic fibrous nanosilica. This is his invention, DPNS, and this actually has attracted worldwide attention, and people from all over the world are using this molecule for preparing various kinds of structures. Okay, it has applications like nanotraps for carbon dioxide capture, CO2 to fuel and cement at room temperature. They have the sand and beach convert CO2 to fuel, black gold as artificial tree, nano sponges of solid acids. These are great implications for the climate change, how to control the climate, and that is the and then convert carbon dioxide into fuel. So these are important applications, and you can see how the department has moved from one area to another area, continues to move from one area to another area to be on the forefront and contribute to the development on the fundamental side on one side and on the applications on the other side. And this is Ravi Venkataman. He's a computational chemist. So he actually develops what is called as the protein charge transfer spectra to interpret these spectra. Computational techniques are used, and electronic molecular breadboards, okay, quantifying protein dynamics. These are all very in detail. They are themselves in detailed topics, so you cannot go into all of those details there. So molecular dynamic simulations, electronic structures, charge transport between molecules, and therefore these are all at the forefront, and a lot of effort is required, and this is one, it requires a lot of computational effort. Sophisticated computers are required for this, and today of course computation has become a keyword in many of the research areas, especially with artificial intelligence, the deep learning, things like that. That has become even more important. Yes, so this is probably the last one, and we have quantum chemistry group. Vamsee is a relatively young man, probably 1 or 2 years old. I mean, joining here. The group is developing quantum chemistry tools to understand the nature of charge transport in organic materials, intermolecular interactions where fundamental van der Waals scaling laws are not applicable, the nature of core electronic states, how they are influenced by weak van der Waals interactions. Understanding of these will develop better materials for solar energy capture and energy storage. Okay. So the department has been organizing workshops at regular intervals, schools at regular intervals. This is a whole list of those, which is yeah, of course you cannot read this, but I just say that okay, there are so many. This is only a partial list. This is still not complete. Whatever I could put in one slide in reasonable resolution, I put them here. Okay, so with that I will stop. So therefore the trend has been like this, that you keep developing, you keep moving, your fronts keep moving, recruit people accordingly, and continue to do research at the front line all the time. That has been the main aim. So with that, I thank you for your attention.